Good evening, everyone. Can you hear me okay? Great,
Yeah.
great. Sorry, I was working without a mouse tonight, just my keypad on the laptop, so I'm a little slower. All right, so we're gonna get started. I have a few things I'm gonna touch on before we turn it over to our guest speaker, Matthew Carberry, who I see there. Hi, Matthew. Welcome tonight.
Thank you.
You're welcome. You're welcome. I'm gonna go ahead and share my screen. So give me about 5 minutes or so, and then I will turn it over to you. I love all the chatter in the chat, but let's hold off for now and let's use that for questions for Matthew or me at this point. Okay. Okay. All right. So welcome. Many of you have been here before. We're excited that you're here. If you have not been here before, though, just so you know, you are in the Virtual Science Club tonight. The Virtual Science Club is hosted by the University of Tennessee and the Southern Appalachian Science and Engineering Fair, which I'll talk about here in just a moment. But the Virtual Science Club meets 2 times every month, and it's always a Tuesday, and it's always at 7 o'clock Eastern or 6 Central for some of you. The first meeting each month is a breakout session where you actually get to sit down with our graduate student mentors who are specialists in a research area, and you can learn from them about how you can enter a project into the Southern Appalachian Science and Engineering Fair. However, tonight what you're at is our guest speaker session. So that's the second meeting of each month, and so we're going to turn it over to a guest speaker for the rest of this session in just a few minutes. So Virtual Science Club, it's for grades 6 through 12 students, and STEM teachers and leaders are also welcome to attend. It's free. You didn't have to pay anything to come tonight, so free to attend. And keep in mind, the next meeting is 2 weeks from today, November the 9th, and that's a Tuesday night at 7 o'clock Eastern, 6 Central. All right, the Southern Appalachian Science and Engineering Fair, or SASEF for short, that is for just these 23 East Tennessee counties. So what I'm about to talk about is just for these counties. If you are interested in being a part of a similar fair across the state of Tennessee, but you're not in one of these counties, you can let us know. You can reach out to us and we can try to connect you with the correct fair. So SASEF, it's a science and engineering fair that's affiliated with the International Science and Engineering Fair. So if you participate in this fair and you're a high school student, you can actually advance to the international fair. The SASEF dates are, well, right now registration is open, so you can go ahead and register. We do encourage teachers and students to register early because the system helps to remind you about deadlines so you can stay on track to enter the fair on time. There's a pre-approval deadline. That means that if your project has some sort of animal subject or human subject, or maybe it's something sort of dangerous like a flame or something like that, fire, you do need to get that pre-approved ahead of time before you start your project, and that's the deadline to get that done. And then lastly, your final project deadline, all your information has to be completed on your project account online by February 28th, so the last day of February. And the event itself, I'll talk about here soon, is a hybrid event, meaning in-person or virtual, on March 29th, and that's a Tuesday. So if you want to learn more about registering, scan that QR code. That'll take you to the registration page and kind of walk you through the steps that you need to complete. All right, I see a lot in the chat. Do we have an issue with audio? Is anyone having problems?
Someone named Maya Cole can't hear you.
Yeah, but I can hear you perfectly fine.
Okay, all right.
Yeah, me too.
Okay, good. All right, I'm going to continue. Okay, so SASEF is going to happen on March the 29th. That's a Tuesday. It's going to happen in that building right there. It's the Student Union, and that's on the University of Tennessee's campus. And it's going to happen in person, traditionally like it's been done in the past. That's the plan right now. is to do the event in person. Like you'll stand in front of your project, you'll have your project board or your project poster, and you'll have judges walk around and ask you questions about the project you've done. However, this year we will allow students to participate virtually if they'd like to, so that is an option. And then on March 31st, 2 days later, we'll do an award ceremony in this room here, the Cox Auditorium. That's also at the University of Tennessee campus. And it'll also be a hybrid event where you can attend in person and sit in those seats right there, or you can log on and watch online virtually. There'll be an option for either. All right, and I can answer some more questions about SASEF, but really right now tonight, I want to turn it over to our guest speaker. His name is Matthew Carberry. He's a marine biologist and president and founder of Sustainable Aquatics. And something interesting about Matthew is he competed in SASEF when he was a student. So he's been through it. He's done this event, and he can speak more on that too. He's going to talk about what it's like to work at a fish hatchery, plus his experiences as a student of science. So thanks, Matthew, and take it away.
All right, thank you. Okay, everybody can hear me and see me. Is that working okay?
I can't see anything, but I can hear you.
Okay.
I can see.
Okay, great. Well, thank you for inviting me to participate in the Virtual Science Club meeting. I think it's a really great way for all of us to be together without the need for travel. So hopefully it gives us a chance to use this technology to share some exciting science. So as Jason said, my name is Matt Carberry. I'm the president of Sustainable Aquatics, and that's a fish farm in East Tennessee. We're in Jefferson City, so we're about 20 miles east of Knoxville. And as Jason said, I participated in the science fair as a high school student, working with Jan Coley, my high school science teacher at the time. And I ended up attending the University of Tennessee in Knoxville. And as I was attending the university, I was a hobbyist, keeping fish at home in my parents' house, and turned that into a business. So when I graduated, that became Sustainable Aquatics, the business that I run now. My projects in high school were concentrating on sea urchins and sea urchin larvae, sea urchin embryology. So coming full circle now, just today I spoke with Dr. Stephen Watts at the University of Alabama in Birmingham, and we are working together on some sea urchin projects. So that's really exciting how more than 20 years has passed, but coming full circle into somebody who's an expert in sea urchins, now working with my hatchery to advance technologies and the knowledge on sea urchin science and production. So that's kind of an exciting story for me personally, but also a story of how the science fair can lead someone to a career that lasts a long time and is very fulfilling. So I have some slides here. I'd like to share the screen, if that's okay. Okay, is that working? Is that showing for everyone? Great. Thank you for your patience. I'm not as fluent in Zoom as maybe a lot of you are, so just learning.
I can't see the slides.
I can.
Are they visible to most everybody, hopefully working? Okay, that's just the tester slide there. So at Sustainable Aquatics, I was going to give a little background on the company, a little bit about what we do, and then some of the technologies that we use that are based on science, and how we use data to inform what we do. Just like doing a science experiment, one of the really important parts of any experiment is to collect data, and then use that data to make the next steps in maybe further experimentation or conclusions, or in our case, production. So Sustainable Aquatics raises fish. And to do that, we also raise a lot of the fish food. So plankton, phytoplankton and zooplankton. And we make a lot of their food prepared feeds. So pellet feeds and frozen feeds. That's a big part of what we do. What we're known for mostly is probably clownfish. So that's Nemo, if anybody's familiar with the Finding Nemo movies. So that's our poster on the right, where we have 86 different varieties of clowns on that poster. We're working on a new version right now that has over 120 different varieties. Many of them are the same species, but they are different colors and patterns. So much like dogs are the same species, but there's different breeds, we have the same thing with clownfish. So we've done a lot of selective breeding to choose colors and patterns that we want over time. And so over this period of 15 years that the company has been operating, we've developed a lot of different strains of clownfish. So as I said, we're in East Tennessee, we're indoors. Most of the fish that we breed, including clownfish, are tropical. So they need to be kept in a temperature controlled area where it's mostly warm all the time. So it's quite pleasant inside. Some pictures of some tanks here. I'm going to go through some slides and then we'll have some videos as well to kind of give you a better idea. So our core knowledge at Sustainable Aquatics is to create environments for breeding and rearing fish. The main things that we concentrate on are nutrition and water quality. And so there's a lot of science there. We're always learning about the nutritional needs of fish and how we can meet that, both in the ingredients that we put in the food and how we present it to the fish in terms of how we feed them and how big the foods are. And that turns out to be really important. You can imagine that if you have a fish that's breeding, creating eggs, you need to provide good nutrition for that fish so the eggs are very strong. And I'll get into that last point there about growing and processing astaxanthin. That's a photosynthetic pigment that turns out to be a very powerful antioxidant. It's a bright red pigment. It helps fish with their coloration and also with their health. So a little bit of history. This is a pair that was purchased in 1998. So this was before I graduated from high school. And we just collected a nest from this fish, this pair of fish tonight. So they're still spawning over 20 years later. They can live a really long time if we take care of them. So the picture on the left there was taken in about the year 2000. Picture on the right was taken in about 2011. And as I said, tonight we collected from this pair. So just for fun, that's me on the right in about 1998, 99, working in Jan Coley's lab at Jefferson County High School, working on sea urchin embryology. So there I'm checking to see if the embryos are fertile. On the right is a few years later, the very beginning of raising larval fish in a small room in the basement of my parents' home. This is a couple of years later, that same little room where we've put in some shelving and put in some tanks for clownfish. One important thing from the very beginning was good record keeping. So if you notice, there's a Post-it note on each tank, and that has notes on it. So we still do that. I've got a video I'll show you where I can illustrate that we still do that. Every tank has records where we can know how old the fish are, who their parents were, any notes on their care, things like that. And that's really important for record keeping, both for science and for the business that we're running. Another little piece of history on the company. A friend of mine, Dennis Tagrin, was running a plankton farm for almost 20 years, growing phytoplankton and zooplankton. And he came and joined us in 2013. And he retired just a couple of years ago. But during that time, he taught our company how to culture these phytoplankton and zooplankton species. And so we've incorporated them into our production and into some of our projects. So I'll talk about the importance of those in a little bit. One of the main reasons to do the phytoplankton was to raise some of these species that required a more nutritious larval diet. And angelfish is a family of marine fish that have very small larvae. So when they hatch out, they're tiny. They're about 1.2 millimeters. So that's about half as long as a grain of rice. So very, very small. These are some of the species that we were working with. And these three we actually raised all the way through from eggs to adults. And then the middle one, the cream angelfish, that one had never been raised before ever, as far as we know. So we were the first ones to raise that one. Hoping the video is that showing for everybody, the video? Sometimes these embedded videos don't. Great. Okay. So that's a
There.
little video of the tank where we kept some of the angelfish. They're still there, actually. So they're swimming around. Some of them are a little bit aggressive, so they can see me with the camera, and they're kind of coming up to posture and even splash water. So on the left is the silo from the side, and on the right's a video from the top. So this is back to the phytoplankton. As part of the marine food chain or marine food web, it all starts with phytoplankton. These are single-celled algae, single-celled plants that grow using sunlight and nutrients. So we provide sunlight and nutrients, artificial light and nutrients, so they can grow very densely. So we concentrate those. Here's some bottles of phytoplankton in small bottles. Here's some larger plankton reactors. Part of the system, all the water that goes into those has to be sterilized so we don't get blooms of bacteria or algae that we don't want to see. And this is a product that we sell. We use it in-house to grow fish, and we also sell it to hobbyists who keep aquariums who want to feed their corals. So this is a picture just showing how it's used to raise fish. We put the green phytoplankton in the water to tint it green on purpose, and that helps the fish to see their food. And it also serves as an enrichment for the zooplankton that the fish eat. I've got a little bit of information on the zooplankton here as well. Rotifer is a small zooplankton that we grow to feed 2 clownfish. So this is a picture of some reactors where we're growing algae, but then also growing rotifers. The cobepod is another small zooplankton. This one is the species, this one Parvocalinus, that we use to raise angelfish. So you can see on the scale there, that's under the microscope, they're pretty small, about 40 to 60 microns. So you can see that 0.5 there, that's half a millimeter. So very, very small. You can see the eggs and the larvae on the slide there. More pictures of those same copepods. These are a zooplankton, again, that are food for the baby angelfish. So here's where we put the eggs into a tank, and then we're adding both, of course, the eggs are there, and we add the food. On the right side, you can see the angelfish when they're about 6 to 8 weeks old, at that stage in development, and so they look like they're their parents at that point. But at the beginning, of course, they're very small, take a couple of months to get to this stage. So again, that was the first time that species had ever been raised before. Here's a picture. This is dottyback larvae, but also very small, and it gives an idea of what they look like when they first hatch out. They're clear, almost clear. I don't know if you could see in the right side of the photo against the black background, you can see some little dots in addition to the fish, and those are rotifers. So it gives an idea of scale of the size of the rotifer versus the size of the fish that's eating it. Here they are swimming around hunting. So we've got the kind of a 3-stage food chain happening in this tank where we add phytoplankton, and the rotifers eat the phytoplankton, and then the rotifers are themselves the food for the fish larvae that we have here. So this is just a little timeline of the feeding, what days we're adding which food. So in the very beginning, we're starting with those very small foods, and then over time, we're going to larger and larger foods. The DPH here is days post-hatch. So the days after they hatch is how we're measuring their age. So like with young children, a lot of times you'll hear parents talking about it in terms of how many months old the baby is. With fish, a lot of times we talk about it in DPH, days post-hatch. That's a critical period for a lot of young fish. And then at about 2 months, we transfer them into grow out. So here again is a person for scale, but the little fish there about 2 months old in that cup. And they're going from the larval tanks, which are round, into a glass aquarium. So at this point, they're just a small fish and they require normal aquarium care, and they'll grow from that size to adulthood. And here they are in the tank, the video showing them swimming around. So these are lemon peel angelfish on the top, and then the bottom, that's an African flameback angelfish. Here's a video of them. These are about 3 months old in this video. Here's a little bit older lemon peel angelfish. And those cream angelfish. Very active fish. So we call these a high metabolism fish, meaning they use a lot of energy. And because they use a lot of energy, we have to provide them with a lot of nutritious food. So then the question is, what's next? One of the next things we're working on is tangs. We've raised tangs to about 40 days old before. We haven't raised them all the way. So that's the next challenge that we're working on. The one in the middle, the yellow tang, comes from Hawaii. And that's a big challenge for us because recently, early this year, Hawaii closed all collection of aquarium fish. So that fish is not available except from hatcheries. And right now, the University of Hawaii is breeding some of those fish and making them available, but they're not widely available in the trade right now. So that's something we're trying to work on. They closed it due to conservation concerns. So the fish that we have now are really the only ones that we can get, and we're working to spawn those. And I have a short video of the tank that we're working on to rear those. So here's an outside video. You can see how tall the tanks are. These are fish that spawn in the open water and release their eggs into the water column. So they need a large vertical distance to do that spawning dance. Unlike clownfish, as you saw earlier, that'll spawn on a substrate. So they spawn in a flower pot or on a piece of tile or on a rock. Here's some purple tangs. They're similar to the yellow tangs. They're in the same genus. They're just a different species. These ones come from the Red Sea. You can see blue tangs on the left and yellow tangs on the right. So this is from some of the first work that we did. You can see in the upper left, those are newly fertilized eggs. Those are about 12 hours post-fertilization. So they're not hatched yet. And then you can see the other pictures show the development through about 40 days old. So here's blue tangs, the same idea, blue tangs on the left. Those are actually babies that we imported from the Solomon Islands, and then their eggs and their larvae. So a lot of microscope work to check on these larvae and see how they're doing and see how healthy the eggs are. So one of the big things about doing this in Tennessee is making salt water. And we use a brand called Instant Ocean, which I used way back in high school and still use now. We buy that salt and it's mixed in these big silos, and then we use it all over campus. We have 7 buildings that we transfer water between. So on the lower right, you can see a picture of a valve and a wristband. So we have those wristbands so that we remember that we're filling the tanks with water. So we don't want to walk away or go to lunch while we're filling water and have a big mess. So that's one of those little reminders we use to keep ourselves from causing problems. This is a filter that we developed. It's based on wastewater treatment technology. It's a biofilter that allows bacteria to grow on it, and those bacteria process the ammonia that the fish produce, the waste that the fish produce, into less toxic molecules that aren't bothersome to the fish. So on the left is a picture of a filtration area with several of those lined up. Then on the right is a picture of a grow out room where we have clownfish that are 2 months old and older growing up there. Part of the filtration, this is called the protein skimmer, and we use it to remove organics from the water. So it creates a foam, and the bubbles in that foam are charged and causes almost like a static attraction of dirt to the bubbles and it foams it out of the water. So that's one of the technologies that we use to filter salt water. Another picture of the hatchery there. Here's a nest of maroon clownfish. So this is the largest species of clownfish. This pair is also quite old, still spawning. And those eggs are nearly ready to hatch. So you can see the silvery eyes in those eggs. And here's to contrast with that freshly laid eggs that are bright orange. Here they are developing. And here they are almost ready to hatch. Here we did a counting of the eggs to see how many eggs were being produced. So this is, they don't come out that color, of course, that's color coded for easy counting, but that particular nest is 647 eggs. Let's see if I can. Okay, so then here's the newly hatched clownfish. These are actually a couple days old. They're just starting to get, if you can see in the upper left there, that one has a bit of a head stripe showing, and they start to get their stripes. They start with their head stripe, and then they get their middle stripe, and then their tail stripe. Here they are a little bit older. And here they are at 1 month old. So this is a glass aquarium. I just used this picture to show the difference between how we raise clownfish and some of the other fish. Where we might raise them in a tub like this, that some of the delicate fish are so delicate that they'll get stuck in the corners of a glass aquarium. And so we're using some tubs for that. Here's a view from the top. There's actually clownfish in that one, but they work for a lot of different species of fish. And here are some of the other species that we've worked on. Damselfish, gobies, a fish called the comet, and orchid dottybacks, a bright purple fish that also comes from the Red Sea area. This is 4 species of blennies. They're all in the same genus. They're called fang blennies. And they're demersal spawners, which means they deposit their eggs onto a substrate. And so we raise several species of those. They're colorful, small fish, great for aquariums. And this one is a sea slug. We call it Bergia in the trade. And I use this as an example of how we do kind of underwater gardening. So you might add ladybugs to your garden to eat aphids. Well, people add these sea slugs called Bergia to their tanks to eat a pest anemone that sometimes grows in their reef tank. So in that bottom center picture, you can see on the right side of that picture are those anemones, and then the Bergia little sea slugs eating those. So it's kind of a way of natural cleanup for your aquarium. So we raise those slugs as well. Here's a close-up of that picture. This is the Hematococcus that I was talking about earlier that makes astaxanthin. So this is an algae that grows green, and then in the later stages of its development, it creates a lot of this astaxanthin and turns red. And so we've developed a process to extract that astaxanthin and include it in our food. I see, do you have a question? You want to go ahead and ask a question? Anish, go ahead.
Yeah. So when you said you added sea slugs to the aquarium, just like you added ladybugs to eat aphids, the sea slugs eat the anemones that grow. How do the anemones get there? Because they don't grow. I know anemones are living organisms, so how do they get there in the first place?
That's a good question. So those anemones come in on something that you might add to the aquarium. So let's say you add a rock that has some things growing on it. It might have one of those anemones on it. And it's something we would try to avoid, but sometimes you have a lot of diverse creatures growing on these rocks or on the base of a coral, and it might find its way into your aquarium. Those anemones sometimes can grow really rapidly. So if they grow too quickly, they'll take over, and it's difficult to remove them manually. They can grow by fragmentation. So a little piece of that anemone can break off and grow a new anemone. So sometimes you can become overwhelmed by them. So the sea slug becomes a good method of making sure you get all of those anemones out of the rocks. So Good question. So that's an unanticipated or unwanted addition to the aquarium that we use the sea slug to help control. So here's our hatchery diet that we formulated for clownfish. We use it for clownfish and for a lot of other tropical fish. And they learn by association of us with the food to come to the front of the tank and eat the food. So one of the things we've done is we've leveraged the technology for food to do salmon. So my father, his family is Norwegian, and he's always been interested in helping salmon farmers to grow the salmon more efficiently, to grow them faster, and to have higher yields, which means to have more salmon surviving. We don't want to lose salmon in the process of growing them. That's been a philosophy at SA with the clownfish, and now we're trying to apply it to salmon. So we talked with some hatcheries in Norway, and they provided us some eggs last November to grow and to do trials to demonstrate our ability to manage water quality and nutrition for salmon. So here's the very beginning of that trial. You can see day 0, the eggs, this is as they arrived, and then the growth of the salmon over time. So again here, is there another question?
I see a couple questions out there. What we can do is we can finish up your presentation, Matthew, and then
Okay.
we'll move on to questions at the end.
Okay, just a couple slides left on this, and then we can
Okay.
do some interactive stuff. Okay, so that's the beginning. One of the good things about, or important things about doing a trial like this is data collection. So that's something we've done here, where we're collecting a weekly report of their growth. So we take a sample of 10 salmon each week and weigh them to get an average weight per fish. So we've done this every week now for 47 weeks. So you can see that they're growing here, and we fit a line to this data set to try to predict how they're going to grow in the future. You can see on the right side that they've slowed down a little bit. We had an automatic feeder that was supposed to be delivered at about week 40 that was delayed, and there had been a lot of logistical delays in 2021. So that delayed our ability to feed them as much food as they wanted. And I think we saw a little bit of a decrease in growth with that. But the reason I show this is to show the importance of data collection and recording. And same here with recording water quality. Oh, this is the last slide of the presentation. This is another kind of application of our nutritional technologies. We've made a diet for axolotls, which is a salamander, a neotenous salamander, meaning they remain as a larvae their whole lives. That's been a really fun project as well. Okay, so I have a couple videos, and we have hopefully time for a few questions. Should I start? This salmon video is kind of an introduction. It's about 2 minutes long. Maybe I'll start with that one, and then after that we do some questions. Does that sound okay?
That sounds great. Yeah.
Okay. Can you hear me?
Yeah, we can hear you
Okay.
okay.
Great. Um,
All right, so I think what we can do, Matthew, is we could do questions now if you're up for that. And what we'll do is we'll have the students, there's a little reactions button if you don't know about it. If you click reactions and then raise hand, do that if you have a question, and then I'll call on you. And we'll just go through questions that way. I already see a few popping up.
okay.
All
sure.
right. Preston, if you want to unmute and ask a question.
I'm kind of curious how many phytoplankton are in a cubic meter of water in the phytoplankton tanks.
Well, that's a good question. There are a lot. So we raise them to several thousand cells per milliliter. So there would be more than trillions in a cubic meter of water, a very, very large number, because these are very, very small cells. They're about 2 microns in diameter, 2 to 10 microns in diameter. So probably many trillions in a cubic meter if there's many thousands in a milliliter. But so they're pretty high density.
Thank you.
And Annalie?
I have two questions. One, is it normally only Atlantic salmon, or can you also have more, like, different kinds?
Another great question. So right now we're doing Atlantic salmon. We'd like to try a couple of different species. King salmon is another one we'd like to try. Of course, I think our system would work well for trout in pure freshwater. But yeah, there are several salmon species that are attractive. These are of course grown for food. So we might apply our technology to some other species as well.
Okay. Also, what do you do with the fit? Like, if they get to be big enough and adults and everything, what do you do with them? Do you keep them or do you do something else?
So these are being farmed for food. Of course, the trial that I showed you, the purpose of that is to show that our food can produce healthy salmon. But at the end, those salmon would be harvested for food and processed for people to eat.
Okay. Would it be just people, or could it also be like marine animals?
It could be marine animals, and some of the salmon parts are made into animal feeds as well. The primary purpose is for humans to eat. And with the salmon we're growing, I'm talking to a couple of people about selling them to restaurants. Of course, the crew here is excited to eat salmon as well. But yeah, to restaurants. And then maybe some of the salmon would go into maybe animal feeds.
Okay. Thank you.
Anish?
So you said you study axolotls as well, correct? Because you showed the pictures of them in the slides.
That's right.
So do you, what exactly do you study on axolotls?
So right now what we're looking at is the length of time it takes to go from egg to maturity, which with axolotls right now can take about a year for them to become mature. We're just starting a trial with the University of Kentucky. They have an axolotl program there. Dr. Voss runs that, and we're sending them some food so they can compare our food with the food that they're currently using to see if it helps them to grow to maturity faster. And then at maturity to see if they produce healthier eggs once they start spawning. So those are the things we're looking, it's nutritional that we're looking at right now to see how that affects their health and growth.
Okay.
All right, and I think Roxanne was next.
I don't know how to state this question, but like, what fish have you studied that's the deepest in the ocean?
Good question. These are good questions. The deepest, most of the fish that we breed and that we work with are fairly shallow water fish. So reef fish from tropical reefs. So most of them are not very deep at all. Some of the tangs, I guess, would be the ones that range a little bit deeper, maybe 60 to 100 feet. But most of the fish that we raise are very shallow water fish.
All right, so Spencer, I'll have you ask a question next, and then I see one in the chat too from Zoe. If you can't raise your hand for some reason, feel free to put it in the chat and I'll try to get to it too.
Mr.
Spencer?
Carberry. Oh, yes, sir. I know you're like talking about sustainable aquatics and like acquiring DT's plankton farm, and you mentioned like having multiple like buildings on campus. So I just wanted to hear about like your business model and like how big y'all are and like what it was like getting that all developed.
Sure. So it started out obviously really small. I was selling some fish while I was in college. And then when I graduated, made it into a business. I thought at the beginning that the industry was very small. So I didn't think the business would be able to grow very much. But it turns out it's very large. So there are only about 6 hatcheries in the world that do what SA does. SA is the largest one by production of fish. There are some other ones that are larger by acreage or by number of gallons, but we're the largest by number of fish. So we have about 30 people that work at SA, and we operate 7 days a week, 365 days a year. And that's been an organic growth over time. So we started with our main building, and then we're in the old part of Jefferson City, so there were some buildings next to us that were available. One of them, the neighboring building, Skeen Realty, was located in there, and they offered to move their business so that we could have the one next to ours and extend it, which was really helpful. So we're in a small town, which helps in some ways, has helped to let us grow that way. But yeah, so it's been a long period of organic growth over the last 15 years, from my parents' basement to a commercial facility, and now growing and having departments and managers. And we found it very important to have a structure, both in terms of protocols and management structure, so we all know what we're supposed to do every day. In the beginning, when it was fewer of us, we could all communicate easily between 5 or 6 people, but now we need to make sure we communicate following a structure of what our plan is for each day. And so that's really helped us to continue growing. That's something we're learning all the time as a maturing company.
Matthew, is there anything further you want to get to, or do you want to just keep taking questions? Because we got a lot of raised hands, so
Yeah, I can keep taking questions. I had a couple little
Okay.
videos, but they're more extensions on what's been shown already. So I'm
Okay.
happy to answer questions. They're good so far, I think.
Sure. Yeah, I saw one in the chat from Zoe. I think back when you were talking about selling salmon to restaurants potentially. She said, do you breed your other fish like clownfish or angelfish for retail?
Right. So the primary customer of Sustainable Aquatics would be a retail pet store. So SA raises these aquarium fish, and then we ship them to retail aquarium shops all over the country, and then they sell them to hobbyists. We do ship internationally as well. During COVID, that's been really tough. So we've done more domestic shipping, and thankfully that has been pretty strong. But yeah, so mostly they are sold for pets.
All right, and let's go with Cecilia. You have your hand raised.
Yeah, so you said earlier with Roxanne's question, I think, you were saying that the tangs live in the deepest part of water. So how do you simulate that within your aquarium?
Another good question. So those silos that I showed, those are about 9 feet deep, and that's not very deep, but it's deep enough that it allows them to behave naturally as far as their spawning behavior. So their spawning behavior involves a spiraling dance that goes vertically about 6 feet generally. So they'll start and they'll dance, and then they'll shoot up about 6 feet. So those fish that need that deeper water, we provide a deeper tank for them so they can do that spawning dance, and we get good fertilization of the eggs. Because during that dance, they're releasing eggs and sperm into the water, and then we get fertilization and collect those eggs. So of course, we're not simulating 100 feet deep, but it seems like the 8 or 9 feet deep is enough for them to have those natural behaviors.
Okay,
All right.
thank you.
Sure.
Thanks, Cecilia. I'm going to have Jefferson ask a question next, but before you say anything, Jefferson, I think this is a good one for every student on here. Zoe put in the chat, do you take interns or volunteers? So they're interested in helping, it sounds like.
Sure, we do. So we're right across the street from Carson Newman University, and we've had interns from there. We've had interns from the high school. We've had interns from University of Alabama and Georgia Tech. And we have a biologist with us now that was an intern a couple of years ago. and now is a full-time employee as a biologist. She graduated with her degree in biology. So that's a success story. That's one of the things we like to see. But I think it's been a good experience for interns, both to see how a business operates and to get some biological knowledge, and a good experience and recommendation for wherever they go next in their education or work.
All right, and then her second part of the question was, what's the longest living freshwater fish that you have seen or you have? I guess you have right now.
Okay, freshwater fish. We don't have very many freshwater fish. We have some freshwater fish that are a few years old. On the saltwater side, we have some that are probably over 30 years old. I have a pet batfish. His name is Orby, and he is about 23. So I had him when I was in high school, and we still have him today. On the freshwater side, I don't know how long some of those fish live. On the saltwater side, we're still learning. If you look at aquarium books, it has ages for these fish. It says they might live 5 to 7 years, and we know now that they can live a lot longer. There are also some fish that live seasonally, like killifish, that only live maybe a year or two. So it really does vary a lot.
All right, and then Jefferson, sorry, you've been waiting. What's your question?
How many fish do you produce a year?
A year? That's a good question. I'd have to do some math on that. Let's see.
Well, Matthew's thinking we have about 5 minutes left. I don't think we're going to get to everyone's question, but about 5 more minutes of questions before we start to close out.
Okay, I think we produce about 500,000 fish a year.
Wow.
Yeah, I've never been asked on that timeline before, so it's a good one.
Okay, let's see. I think Aaron's hand has been up a while. Aaron?
This isn't really a very good question, but do you do tours?
We do. We've done tours with student groups and with aquarium groups. We haven't done any recently, obviously, but we do like to invite some of our friends and local schools and things like that to come in and, sorry about that, and do tours.
Thank you.
Sure.
Okay. And let's see. Kim, do you want to ask a question?
I just wanted to remind everyone, if you're very inspired by this and decide you want to do a science fair project involving fish, they are vertebrate animals, and we will need to have vertebrate animal forms filled out for them. So just keep that in mind. You have to submit the forms. Jason gave you the dates earlier. I think, when are the forms due, Jason?
That would be January 18th is the due date.
Okay, so if you're going to do something with fish, you need to get those forms in.
All right, we're going to try to squeeze 2 more questions in. I see, so Anish and Roxanne, I think we've heard from both of you already. So let's do, I just see iPhone, but we'll do iPhone and then Amelia next.
Hi, my name is Reagan, but I had a question. So what do you do if like fish are in infected water, and if like it gets infected like accidentally, what do you do with the fish?
Another good question. So we have a lot of treatments available, treatment options. The best treatment that we have, though, is prevention. So we actually have a separate building where we bring all new fish in. Anything that's not from the hatchery comes into that building, and we do what's called a prophylactic treatment, so preventative treatments, to make sure that we don't bring parasites or pathogens into the hatchery. So there are treatments we can use if the fish get sick, but really we try to prevent any diseases from coming into the hatchery so that they don't cause problems later. And that's something that we pride ourselves on, is having what's called a specific pathogen-free facility. So the main pathogens that we're worried about, we don't have them in the hatchery building. We make sure we don't bring them over. Good question.
Go ahead, Amelia.
Okay, this is not one of the greatest questions ever, but what is the smallest fish you have like ever worked with?
Probably the smallest fish we worked with are gobies. So some of them are fully mature at less than 2 inches long. The interesting thing about those is that when they hatch out, they're almost as big as clownfish. So they're easier to raise. Their mouth is big enough to eat rotifers that we were talking about. But yeah, they don't grow any bigger than about 2 inches long when they're fully grown. I think that's the smallest that we work with.
Thank you.
All right, so we've got about 5 minutes left, and so I'm sorry, there's been a lot of great questions, but we can't stay on all night. So we're gonna cut off questions now. Before I close everything out, I did want to say a big thank you to Matthew for being here. We really appreciate him taking time out of his night. So a round of applause for Matthew. Thank you.